Hello, my name is Ruth Massey and I am a Professor of Microbial Pathogenicity at the University of Bristol. As the name of the title suggests, my group's interest is looking at microorganisms, and these are life forms that consist of bacteria, fungi and viruses. It couldn't really be a more relevant time to do this type of work given that we're living through this coronavirus pandemic. And we have lots of really amazing researchers here at Bristol looking at the virus, how it causes infection, but also looking at our immune system and how we can protect ourselves from this. My group has a slightly different interest, and we want to look at a really important aspect of viral infections, especially when they're in the lung, and that is that often after the virus has caused the initial damage during their infection, bacteria and viruses come in, take advantage of this damage and cause what we call secondary infections. And these are particularly important because once they start to develop in coronavirus patients, the situation is really quite grim and very difficult to deal with. So what we want to do is to develop technology that will indicate to the clinician when that patient, just before or just as soon as a secondary infection is about to start, when that's happening, so that they can treat the patient appropriately. To do this, we're working with two groups of collaborators. The first are the doctors at the two large hospitals we have in Birmingham. Now, because these patients spend quite a lot of time in hospital, with the patient's permissions, the doctors are able to provide us with samples from the nose and the throat and the lungs of these patients daily and weekly, so we can get multiple samples from the same patient. And from this, we're studying the patient's microbiome. And what we mean by that is what microorganisms are in those samples and how many. And this is important because we expect there to be an abrupt change or some sort of shift in this microbiome that will indicate when a secondary infection is about to start. To achieve this, we're working with our other collaborators at the University of Birmingham. And with them, we are going to sequence all the DNA and the RNA in these patients' samples, and then using sort of computer-based bioinformatic tools, we can define the microbiome and look for that over time in each of these patients. Now, Bristol is the perfect place to do this type of work because it has access to all these cutting-edge technologies, but it also has these really close collaborative links with the two hospitals. And together we have this sort of shared goal of turning science into medicine to really help improve patient outcome. The next step for this work is that once we've detected and worked out what these early signals are, is to translate them into a quick and easy test. Preferably we'd like it to be a point of care, and what we mean by point of care is something that can be done at the patient's bedside to give an immediate, quick diagnosis so they can be treated really quickly.